Welcome back to the Computomics podcast. We have another very exciting guest today. Her research focuses on optimizing agricultural processes through integrating molecular biology technologies. And as a professor at the Technical University Munich, she also works on characterizing native biodiversity and identifying its characteristics and developing optimized breeding strategies for complex properties. Schön, welcome to the Computomics podcast.
Thank you very much.
Chris and I had a quick talk before and said it was okay to use first name basis. I'm honored, thank you. And I was wondering, my research showed that you took a sabbatical in 2020 to focus on research. What exactly did you focus on?
Well, we had very promising results from a research project, and we wanted to make the best use of these results. And so the sabbatical was mainly about analyzing it in collaboration with one of my students. And that was a very important part. And then, of course, in research, we're always concerned with what comes next and how are we going to make the best out of the results we have, because research also opens new questions. And so another very important task was to define future research topics.
And what would be those topics that you defined?
We're mainly concerned with biodiversity and how to use it. And so we're trying to find out what is actually useful biodiversity, because many people think that maximum biodiversity is what we really need, but we feel that this is a bit simplified. We really think that we have an optimum biodiversity that we should target, and so that was really the research I was working on, and maybe how do we measure optimum biodiversity.
Did you already have results, or is this something that you're trying to find out in the next research cycle, if you will?
It's something I try to find out in my future research, because I was quite surprised to find, even though we use the term biodiversity in economics, in many aspects, in interdisciplinary research, in breeding, there's not really a very good measure for characterizing biodiversity in a way that you can say this is a good level and this is maybe too much or not enough. So I thought that was quite surprising, and that's something I want to focus on.
Definitely sounds like something that you should have a better understanding for, if that's something that you're trying to strategically achieve, right? Can you give us an inkling of how you're trying to measure it, or a working hypothesis maybe?
Well, I mean, there is some concepts out there where you kind of say, like, I can give you an example maybe from interdisciplinary research. You know, if you have groups that are too diverse, the research might not work because people don't understand each other and they don't know how to talk to each other. Whereas when you're only like a very confined group with a very focused topic, of course, there's not much thinking outside the box. So, I mean, there is some strategies to identify the optimum diversity in groups, and that is what I think might be a concept that I will try to use in breeding also, how to identify the optimum diversity.
And you said in introducing these new goals that you're aiming to go for in your research that you already had some promising results, or results that actually led you in that direction. Can you tell us a little more about those?
Yes, what we found was, well, maybe I'll start with the research question we had. There's I work on maize, and there is landraces, for example, of maize, which are populations that were used as varieties in the 50s here, like in Europe. But the breeding progress has stopped on those populations, because breeding has focused on like hybrid breeding and has not used those populations anymore, only as the initial donor of the diversity that we're working on now. So one hypothesis is that there is a variation in those landraces that we can use for widen the diversity of the germplasm that we're using today. But we don't really have any good evidence for that. It's a hypothesis, but many times, like for example, as an example, a resistance trait, usually like pathogens and the plants co-evolve. So why would an old landrace have a resistance to something has evolved over 50 years, right? So the question really is there useful information and useful diversity in those landraces? And so there hadn't been much evidence for that because it's very difficult to analyze them and to really get a good idea about the diversity contained in the landraces. And so what we did is we focused on very few landraces, not a huge broad spectrum, but just very few, and analyzed them in detail. And we used genomics techniques. We did a lot of sequencing and genotyping. And what was most important, we had very good phenotypic data. So we evaluated a trait that we were interested in, which was early development in many, many environments. And with these data, we could really show that there is genomic regions that contribute new diversity and that is not present in the elite germplasm, and that we have favorable effects of some of these areas in the genome that could improve the early development of today's breeding material. And there's very little evidence out there that has shown this before. So we were quite happy to be able to see that.
And this research you were just describing, is this part of the maize project that is funded by the German Federal Ministry, which you are leading?
Yes, exactly. That's what we started out with, because it really, like everybody talks about all those precious genetic resources that we have in seed banks and that are available for breeding. And why don't we use it? And why do we work with so little diversity in the germplasm and all this? But there was no evidence that the landraces should be useful for quantitative traits. Of course, there had been some examples, like the Green Revolution gene, for example, that is, of course, also came from a genetic resource where you make wheat shorter, and so you can intensify the cultivation. So, of course, that was a very important example where a genetic resource delivered a useful allele or gene. But we work mainly with traits that have a continuous distribution, that are quantitative, coded by many, many genes. And for those quantitative traits, it had not really been convincingly shown. And that's why we set out to make a little progress in that respect.
Could you specify an example, like which quantitative trait you would be looking at or have been looking at, actually?
Mm-hmm. Yes, we were mainly interested in early development of maize, because maize is, of course, a wonderful crop. That's what I find. I love it, and I think it's enormous what this plant can really create in terms of biomass. It's a very healthy plant, but one of the criticisms that some people have with maize is that you plant it very late in the year, and then that the soil is not covered for a long time. You need herbicides to take care, so there's not too many, or the weeds, that there's not so many weeds. And and also you have like a leakage of nitrogen if you don't have a good soil coverage. So this, and then also if you plant maize earlier, you can extend the vegetation period.
What are you trying by looking at this quantitative trait of, could we say, the point in time where you actually plant it? Would that be a correct way to describe it?
Well, the point in time we plant it is how the farmer, that's determined by the farmer. Mm-hmm. What we're looking at is especially if the point in time is very early and the temperature is very low, because usually in April, temperatures in Central Europe are not very high. Is there maize types out there that develop much faster than others, that maybe don't need so much temperature, so much heat to really develop fast? And that's the idea.
And you've made progress in identifying these types or land races of maize in the maize project?
Mm-hmm. Oh, yes. So what we could actually show is, of course, in our experiments, we always have checks that we use as the gold standard and to which we compare what we have out there. And so what we did is we planted 1,000 different maize types, genotypes we call them. And so we planted 1,000 of them. And so what we could actually find is that some of them develop much faster than the checks, the commercial varieties that we had out there. So that was already the first success that we had. And then what was really nice was that we could identify genomic regions that were responsible for this response. And then the third step was that we could show that some of these of the variation contained in these genomic regions was not present in the elite germplasm. So that was really what we wanted to do and what we set out to do, and what was very nice to show.
And which methods did you use? Because from what I understand, you have this kind of novel way of including different data sets in your analysis as well.
That's correct. And I have to say, of course, it's not only my group that works on this project. It's 10 partners. And so it's always a collaborative effort to do something like this. Well, first of all, it's very difficult to generate these plants that you can test in the field repeatedly. And we are collaborating with a private partner to work on this, and they generated the material that would have not been possible at a university, because it needs huge facilities to do this in a short time, because time is also very important in the breeding context. So that was the first thing that was important. And then we also collaborated with partners that are experts in bioinformatics that generated reference sequences from the material that we worked with. There is a maize reference sequence, but it's based on US material. And so the European material, what we call flint material, is so different from the US material that we needed our own reference sequences. So that was another project, and that was a basis for the analyses. And then, of course, we also had big field trials that were distributed across many locations. And then we collaborated with the group in Hohenheim, for example, or we collaborated with a group in Spain to also have our field trials there, and to get very good phenotypic data.
You mentioned that you looked at the flint lines. This was in the first phase of the project. Mm-hmm. Are you focusing on the other lines? I think they're called dent lines in the second part. And can you specify what the difference is? Because maybe I should have said that in the introduction. I am not from the field, so I always ask from the perspective of someone who doesn't really know a lot about it, but is excited to learn more. So how would you explain the difference between the two and the focus in the second phase?
The difference between like the flint lines and the dent lines originate really from hybrid breeding. In hybrid breeding, what you do, actually you do something that's quite funny. You have, let's say, outcrossing populations, and you have several of them. And then what you do is you inbreed those populations by selfing them, and you create a lot of inbreeding depression. And then what this does, though, is that you get immortal and reproducible genetic units, because inbred lines can be multiplied many times. And it helps to select the best plant out of the population also. So that's the two ideas behind making things bad. And then in the end, you cross again 2 inbred lines to make the hybrid. And so the reason why we have like a flint and a dent population is that the more diverse these populations are, the more they complement each other and the higher the heterosis usually is, which gives us additional advantage in hybrid breeding. And traditionally in Europe, hybrid breeding started in the 50s, and the material came from the US, and it was material that was adapted to the climatic conditions there, and it had high yield. It was very high yielding, but it was not adapted to European conditions, and the populations that we work with, the landraces, they were adapted, those flint landraces. And then people made inbred lines in each of the populations. And so the flint then brought like the more cold tolerance and early flowering and everything that dent from the US didn't have. And the two populations, they're called dent and flint because the flint has just very hard kernels and it's kind of a flinty type of kernel. And the dent, it looks like a tooth, a little bit like a tooth, and that's why it's called dent germplasm.
That's good to remember it. So what exactly are you trying to do in phase 2 now with, I guess, more of a focus on the dent lines? How do you put the research results from the first phase together with the second phase?
I know I should focus on the dent lines, but I'd like to mention one thing.
Of course, please.
We're still working. I mean, the material we developed during the first phase in the flint, it's very, very useful for now cloning a lot of genes. It's like we can crank out a lot of variation and really identify now the genes that are underlying a lot of traits. And so that's what we're concerned with also in the second phase, and we work on that. And I think we're quite happy with, we have variation for more than 50 traits. And I think that gives us a lot of potential for like identifying genes that underlie genetic variation in maize flint material. And in the dent material, we felt that it's not enough just to focus on one of the European pools, but we're now looking at dent material that we generated because we thought also that drought tolerance is a very important topic in the future. Like a lot of areas in Europe already suffer from drought, and we thought we would like to also address drought tolerance as a very important trait for the future. And so we decided that we will take 8 lines from the dent pool, and these 8 lines had been pre-tested in another project where like a very large set of 250 lines was tested in 30 environments all across Europe and under a lot of drought and heat, in a lot of environments that varied for drought and heat. And so we selected 8 of these lines from which we think that they will give us variation for drought tolerance and that are also adapted more to the Central European environments. And that is what we will now look at. We derived inbred lines from this material, and we'll test that now in Hungary, in Italy, and also here in Germany, and investigate the drought tolerance, and then also try to, of course, to find the genes underlying the drought tolerance.
And now you raised my curiosity when you mentioned you identified 50 traits from the flint lines. Can you go into a little more detail there? Maybe ones that you personally find the most interesting or that get you excited the most.
Well, I think what's quite exciting is that there's like photosynthesis related traits that we could look at because the material varies a lot. I think I have to say that one of the drawbacks is that we always find genes that are bad, the alleles that are bad, but that doesn't matter, because once we find the gene, we can go into the material, to the elite material, and then screen for allelic variation there. So the landraces kind of give us an idea on which genes are involved, and then we will look into the elite material and see how these genes are, how they look, how the alleles look in elite material there. So that will be very useful. And then, so it's photosynthesis related traits, it's disease resistant traits, it's developmental traits, like, of course, also the plant morphology is very different. And then, of course, we also have, of course, yield data, we have maturity data, we have flowering time data, all the agronomic traits, plant height. So we have a lot of traits assessed in our trials. That's and all of them are very exciting, I think.
You don't have a favorite?
I think I will probably, what we're doing at the moment is we're, two of my students one works on lodging, that is the plants fall over because of some root traits. So I think he picked the hardest traits you can go for, actually. So, but he needs a challenge, I suppose. And I think, and another one works on early development now, on a gene about early development. And I think probably if we think about going into the next phase, we might actually go for photosynthesis-related traits.
Why those? If I made you pick a favorite, I know you didn't choose to,
yes.
But why those?
I mean, photosynthesis in C4 is still not understood very well in C4 plants. So maize is a C4 plant. It has a different photosynthesis pathway than like wheat, and it's quite efficient because of that. But the like water use efficiency and photosynthesis, of course, are related. And so I think that's why I'm interested in it, because the accumulation of biomass, of course, is something that also has a high relevance for practical cultivation of plants. Mm-hmm. I think I like it because it's a combination of basic research and with a high impact. I think that's why I like it.
Yeah, makes total sense. And I mean, we've been talking a lot about the research, both the results you've had and also the goals. But there's also a technical aspect to the maize project, if I understand that correctly. What would you say are maybe the technical goals or things you've already achieved? Let's focus on the results first. Have you any methods or technical advancements that came from the project?
I mean, actually, we have quite a few. Well, first of all, what I already mentioned is that we analyzed 4 reference genomes, or kind of assembled 4 reference genomes for the European maize. That is very important for all the cloning activities that we have. And that's not trivial, you know. I mean, to assemble these genomes it's quite complicated. And then what we also, of course, have achieved is that we have developed software and methods, statistical methods for how to analyze the data, how to compare haplotypes in the landraces to haplotypes in elite germplasm. So haplotypes are short DNA sequences that we compare. And then we say, okay, exactly that sequence is present in the elite germplasm or not. But the definition of a haplotype, how long it has to be, how many bases does it have to contain, is very, very difficult. And it's still an open question how to do that. And I think we've done very well on that. And then a colleague in Göttingen, also from the maize project, developed some software called Haploblocker to define haplotypes in a slightly different way than how it's done in human genetics. So I think we have also made quite some progress there. Other colleagues have worked on implementing deep learning approaches or neural networks for predicting the performance of like maize lines. And I think also some progress has been made there. And I think actually for us also, the generation of the material in itself was a big success, that it actually worked to get inbred lines out of landraces, because you have to see those landraces, they carry what we say a high genetic load. when you inbreed them, they will very often die. So because they have such a high load of genes that are unfavorable and or even lethal, and because they have not gone through a breeding process. So that's also been quite good for us.
Just the length of your answer shows how much you've already achieved with the project. In an ideal world, what would you say, or what would you dream the outcome of the project to be?
I think I would like it if people used the resource, because we made it publicly available. And I think there's a lot more work than we can shoulder, even if we're 10 partners. There is so much variation in there, and I would be very happy if colleagues would kind of pick up some of the lines or like some research questions, and we are very happy to share the material with them.
Where would someone be able to access that? I mean, we have all these listeners out there. Someone might now be curious now that you've mentioned it. Where can they find that information?
A lot of the data has become public already through publication. So you can either download it from through the publications, or a lot of the material is accessible through material transfer agreements, because they're genetic resources, and we're obliged to make them freely available to other people, and we're happy to do so, very happy to do so. And of course, then also, depending on the research topic, we can also make the genotypic data or sequencing data also available to partners.
Is there an aspect, we're heading towards the end, but I always like to ask, is there an aspect that we haven't talked about that you personally find noteworthy about the project?
I think what I find noteworthy is that people realize that this is a long process. Those populations haven't seen breeding for a long time, right? They're like 50 years back. And so what we need to do is close the gap in performance between those populations and what we have now. That's one thing. And that cannot be done in a very short project, right? So that's a very big task. The second thing is maybe to start out with, or to end with what I started out with, that diversity by itself is not a favorable, beneficial thing. You really need to know what kind of diversity you want, how much diversity you want, and how to make best use of it. I think that's really something that is important. I think what's also important to me is that the project is a very good example of a public-private partnership, I think, because we're able to publish all the results that we obtain, that we can share all the material that has been provided, and that also that the private partner, KWS, has made a huge effort in providing us with phenotypic data. So I think phenotypic data is really a very, very important aspect, and this would not have been possible without a public-private partnership. I think that's also something that's important to me. Yeah, and of course, it's a collaborative effort. Things like that cannot be done by individual groups, and we're very grateful, of course, to the BMBF and that they funded this project, and we've been very happy with the project, yes.
You've achieved a lot. How long will the project run? You're in phase 2. Phase 1 is already complete. Phase 2 will run, if you have a broad idea.
Oh, I think we're not even midway of the second 3 years, I suppose. I'm not very good with these.
That would mean 2023.
Yeah, I think so. I'm not quite sure when it ends, to be honest, because that's not the categories we think in. We're just doing our work, and then, but we're already getting prepared for preparing the next proposal and the next phase, and we already have ideas on how to do that. So it's a continuous process. So sorry, I'm not quite sure, but I think we started like 1.5 years ago. So we should be probably midway in like another 1.5 years probably.
Mm-hmm.
Yeah.
Yeah, so that would be early 2023.
Yes,
I think, but I mean, this goes with everything that you've been saying as well. This is not a process that has a clear beginning. You know, it starts on that date and then it ends at exactly that date, but rather it's a continuous process, and hopefully we'll have even more great results to work with, and then to go deeper, right, in different aspects.
That's the idea, yes.
So yeah, I think we've learned a lot about the maize project. Computomics is very excited to be a part of it in phase 2 as well. Thank you so much, Chris, for this lovely interview, for teaching us so much about the results, but also the goals and the further outlook of the maize project and that particular research. To you listeners, I hope you found this as exciting as I did. You can find some notes on our website, and I look forward to having you all back for the next episode of the Computomics podcast.